Barry, that was terrific. Thank you very, very much. We're going to move into session 3 now, but maybe everybody should just stand up in your space and stretch for a moment. You might be just tired of sitting. Stretch, twist, reach for the stars. Please.
You said that gender is not a disease. I think when my son turned 13, it was a disease. That's cultural. Someone calculated the LD50 of testosterone.
Great. Well, session 3 is entitled Nanomedical Structures and Devices in Development Today, and we have 4 outstanding speakers on this subject. I'd like each speaker, after their presentation, to please take a seat at the panelist table, and at the conclusion of the 4 presentations, we'll open the floor to questions and answers, and thereafter have a very well-deserved coffee and carbohydrate break. Our first presenter is Brian Helmke. Dr. Helmke earned a PhD in bioengineering from the University of California at San Diego and is currently an assistant professor of biomedical engineering, in addition to being a board member of Nanoscale Quantum Engineering Science and Technology Institute and a member of the Robert M. Byrne Cardiovascular Research Center and Cancer Institute, all at the University of Virginia. Dr. Helmke's topic is entitled Nano-Based Devices for Engineering Cellular Life and Death. Please join me in wishing a warm welcome to Dr. Brian Helmke.
Well, thank you for that kind introduction and for the invitation to talk to you today. I'd like to, there we go, title slide. I'd like to put in a plug for my institution too, which is the University of Virginia, and the Biomedical Engineering Department there, which is rapidly growing and attracting new faculty, and point out of interest to this audience that we're working closely with the Coulter Foundation as a partner in their translational research program, one of 9 BME departments that heading this up. And this funding in the department is geared entirely towards promoting translational research, which requires collaborations between engineers and active clinicians. And in the process, we've pulled in interest from the Entrepreneurship Center, and one of the co-directors is the director of the Entrepreneurship Center in the Darden School of Business as well. So it's a cross-school, university-wide initiative in translational medicine, of which nano plays a big role, predictably. So, from my point of view, I'd like to pose what I called the grand challenge, which I think it fits well this session and perhaps the next session as well, which is that we would like to think about using nanomedicine at the cellular level to intervene, to design biochemical or mechanical properties and to engineer the sensing and behavior of cells. And the key here is that at the root of these diseases is a dysfunction in cellular behavior. And in order to engineer those behaviors, we have to first understand mechanism from the cell biology point of view. So this diagram shows here several areas that we've heard a little bit about, touched on in terms of genetic manipulation, and we're going to hear later about reactive oxygen and perhaps nitric oxide signaling and so on. And from the standpoint of nanostructured materials, we'd like to control this interface right here. That is the interface between the cell and its external environment. And one of the goals that's strong in my lab is trying to understand how the cells sense that external environment. And so the goal here is to engineer from a systems biology or systems bioengineering point of view the behavior in very complex signaling mechanisms that arise with an origin at this interface with the cell, between the cell and its environment. So there are a number of fabrication techniques that we can use to manipulate that environment, none of which are terribly new from a material science perspective, but I'd like to make the point that in order to engineer the behavior at the multicellular or tissue scale, one has to be able to fabricate nanostructures reproducibly and robustly enough to be able to manipulate behaviors of cells that can communicate with each other. A single nanostructure or writing your name in nanodots in order to program the cell shape doesn't control the behavior of a tissue. And so one strategy that we've used to do this is in a substrate in which we anodized aluminum and then expanded the pores down to a layer of gold at the base so the pores were in contact with the gold, then electroplated the gold and etched back to create nano islands of gold. And then one can do thiol chemistry on the gold nano islands. And the advantage of this system is that blocking cell interactions with the other part of the material is a non-problem since proteins and hence cells don't adhere to the alumina part of the surface. Another strategy depends on some of the polymer nanostructures that we've heard a little bit about. And so we've worked with Robert Hull in a MRSEC center at UVA using his FIB, his focused ion beam, to engineer holes which we then cast stamps from in polymeric materials. This was in PMMA. In which we wrote the holes. And the advantage here was that in PMMA we were able to write holes at a speed that allows us to pattern at this length scale over a total scale of around a couple of millimeters square. Again, thinking about manipulating environments of cells and not just single cells. And then one can do direct nanocontact printing with those polymer stamps to create arrays of nanodots, or at least near nanodots, in extracellular matrix protein. And the goal here is to mimic these cellular interactions with the material during formation of individual nation adhesion complexes. And the advantage now that we're going after is that we would like to control the surface in a way so that the cell is no longer allowed to remodel and reorganize its adhesions in a way that it normally would in contact with a biomaterial or with its native matrix, so that we can ask questions about what are the signaling mechanisms or the basic biology mechanisms that evolve in response to those remodeling events. So let me give one example on the engineering cell health side, and that is in vascular biology, which is my major area of interest. And clinically, in vascular biology, in response to a stenosis or in atherosclerosis, for example, one strategy is to go in with the rotor-rooter and then put in a wire mesh or a stent to hold the vessel open afterwards. And of course, the problem here historically is that after scraping the heck out of the inside of the vessel and then putting in a metal wire mesh, that the cells in the artery wall tend to overgrow to compensate for the injury, and the stent and the artery recloses or restenoses. And so one way around that is to include in these stents a number of strategies that try to limit cell overgrowth. Now, the problem here is that the cells that are growing in that case are the smooth muscle cells responsible for arterial contractility that are in the layer of smooth muscle cells nearest the blood vessel lumen. The problem is that the endothelium, which forms the normal antithrombotic interface with blood, is gone. The rotor-rooter has taken care of that. And so in preventing the overgrowth of the smooth muscles using either drug elution or various polymer coatings, we're also preventing re-endothelialization of that wound, and it's the endothelium that's critical for preventing future thrombotic events in the patients, or at least it strongly correlates clinically. So again, a number of strategies have been tried here. What we wanted to understand is what's controlling the endothelial migration and the monolayer formation, the tissue formation in these vessels after an endothelial injury. And the strategy that we're using to understand how cells are interacting with each other both mechanically and chemically, is to put endothelial cells on nanotipped pillar surfaces, right now fabricated on silicon, because this is a culture system and it's easy to be reproducible on silicon. But we're working with Kurt Kolazinski at Westchester University to fabricate these. He's a chemist that fabricates these, and he also has these arrays in titania surfaces. So what's new here? So from the bioengineering or cellular engineering point of view, what do you do first? You put cells on a surface of your favorite nanostructure, and then you see what happens, right? And what we noticed here, labeling in these cells, we're labeling nuclei in blue, we're labeling actin cytoskeleton, the major contractile cytoskeleton component of the endothelium that's responsible for cell crawling, for example, and for the tension, we think, between cells in the monolayer tissue, is labeled here in green. And these are two optical sections taken from different height in the cells. Near that pillar substrate, it's a little hard to see in this light, but the arrows are pointing out gaps or holes in the structure where the cell has morphed around the pillars. Not terribly surprising. This has been shown on topography surfaces in the past as well. And at different optical sections, even near the apical surface of the cell, that is near the luminal surface that's normally in contact with blood, we see even here that there are locations of holes puncturing through nuclei even. Okay, so, and to maybe be a little bit more convincing, we have a movie of a Z-stack scrolling through the cell, and we can pick out locations where the holes are consistent through the Z-stack, for example, demonstrating that these are in fact where the pillars are located and not simply variations in structure. So this suggests that in studies on nanotopography, for example, to improve cell adhesion to a biomaterial, that the cell adhesion strength is indeed improved because the cells are interacting with the material by flowing or morphing or adapting their structure to interact with a rough material. And in the presence, in this case, of flowing blood, that would make the cell more hydrodynamically resistant to de-adhesion from the material, from the implant material. The question is, does that correspond to an improvement in endothelial function? Clearly not, because the cells never come together and form a functional healed monolayer the way that they ought to in the vessel. So here's some evidence that points towards why we think. If we look at a confluent endothelium, this means an endothelium that has formed a monolayer with normal junctions and is communicating normally both mechanically and chemically, on these nanopillars, we find now a normal actin cytoskeletal structure, which includes these thick stress fibers in the basal area of the cytoplasm, as well as actin bundles along the edges of cells, and those are associated with the mechanical junctions between cells. And this endothelial layer looks no different than an endothelial layer on a glass cover slip. There's no evidence that these cells are on pillars, at least morphologically. For comparison, as a control here, we used a flat silicon substrate just to demonstrate that a monolayer never forms on flat silicon, as you might expect. The topography is required, as has been demonstrated in the past. In addition, if we label vascular endothelial cadherin, one of the proteins involved in adherence junctions or mechanical junctions between cells that are responsible for endothelial permeability or maintenance of non-permeability to macromolecules, then we find that in subconfluent layers on the pillars where the pillar holes were present, that this is disorganized as you might expect, but in a confluent layer on the pillars, it lines up nicely along cell edges, forming monolayer junctions that look no different than cells in a healthy functional monolayer. If, however, we treat the cells with blebistatin, which inhibits the ATPase activity of myosin, responsible for the contractility in that actin cytoskeleton. In other words, responsible for generating intracellular tension to pull against neighbors and substrate. Then the cells on pillars sink back down around the pillars, the actin becomes disorganized around the pillars, and it's much more evident on pillars than on a cover slip, for example. And in addition, the cadherin structure becomes disorganized instead of lining up normally along cell-cell junctions. So that suggests now that the maintenance of tension between cells through an intact cytoskeleton and junctions is required for maintaining the monolayer on top of those pillars. One other possibility is that the cells generate their own extracellular matrix and their own adhesion to protein matrix on the pillars, and therefore that's just mechanically supporting the monolayer and the pillars have nothing to do with it. We demonstrate here that that's not the case. In cover slips, extracellular matrix proteins fibronectin, for example, is organized into thick fibril bundles that communicate from cell to cell, and adhesions, shown here by the protein vinculin, form short streak-like structures characteristic with traction against the substrate. Whereas on the pillars, the streaks in the focal adhesion proteins are largely missing, and the fibronectin matrix is never quite assembled normally. This means that the mechanical environment between cells is regulated differently than the traction against the substrate, and it's the mechanical tension between cells that's promoting monolayer formation and maturation, not traction against the substrate or cell migration per se. And if we treat these cells, again, with blebbistatin to disrupt myosin ATPase, we find that the extracellular matrix structure is a bit more dysregulated, but it was already disrupted in the first place on the pillars. Okay, so this now suggests a model for engineering cell behavior, for engineering a healthy monolayer that requires using a nano substrate on a length scale where we can ask questions about cell-cell interactions over the length scale of a community of cells that's operating as a communicating tissue, and not just the single cell level to ask questions about cell migration and division and so on. Okay, so I said engineering cell life and death. Here's the death side of the story. And this is the part that's maybe not so surprising to those working with nanoparticles. We assume that they're toxic and kill cells, right? The question is, can we control that? In a separate project working with Qi Sheng and Paul Reed in radiation oncology at UVA, we came up with an idea to address a problem that they face in the radiation oncology clinic. And that is that in response to X-ray radiation, there are certain in our case, we chose lung and liver tumors to focus on that are non-resectable and radio-resistant, so deep-seated solid organ tumors. And these guys can calculate the physics in a way that, that controls the radiation dosing spatially quite precisely to the tumor, but they still see some toxicity to normal tissue in their dosing. So one strategy in the clinic to address this is to try and use drugs that are called radiosensitizers. And the idea is that as the patient receives multiple fractions of radiation, that in the presence of a radiosensitizer, this is basically a chemotherapeutic drug that sensitizes the tumor cells to ionizing radiation by mechanisms that are sometimes known and sometimes completely unknown. So in thinking about this problem, the problem I'm going to show you, I don't think I have to introduce quantum dots here. Quantum dots, as we heard earlier, are nanocrystals that can be coated with, capped to prevent ion leakage or leaching, and can be coated with polyethylene glycol or other polymers to improve water solubility and biocompatibility, and perhaps conjugated to functional molecules. So another piece of the story here is photosensitization. So photodynamic therapy is currently approved with drugs like photofrin made by Wyeth Ayerst to absorb in the clinic red light and emit singlet oxygen that's cytotoxic. The problem here is that because of red light scattering in tissue, this only works on superficial tumors because you can't get the excitation light to the deep tumors. So we thought instead of just using a chemotherapeutic drug to sensitize the cells to ionizing radiation, could we act directly by taking advantage of the photodynamic therapy mechanism, that is triplet energy transfer between molecular oxygen and singlet oxygen that's cytotoxic, by using the quantum dots as the illuminator in the radiation oncology clinic. And the idea here is that in response to therapeutic doses of X-ray, not diagnostic, therapeutic doses of X-ray, the quantum dots will emit enough light to, that if they're conjugated to photofrin, there's an energy transfer to the photofrin that then sensitizes it to catalyze triplet energy transfer and emit singlet oxygen. And we have a paper in review that does all the physics calculations behind this. Now, I should emphasize that we're just taking the simplistic approach here because we wanted to see if the idea worked. We're using off-the-shelf quantum dots that are pegylated quantum dots from Evident Technologies. We're using photofrin, which is already one of 2 clinically approved photosensitizers for photodynamic therapy because we wanted to keep the story simple. But I'm very interested in several of the other ideas that were presented on customizing this for making it more effective. Okay, so in response to 6 megavolt X-ray, the quantum dots do indeed emit light. That's measurable dose. And in the conjugated drug, that emission is largely quenched, showing that the quantum dots are acting as an energy donor to the photofrin. As shown earlier, difficult to see here, the green spots in cells are quantum dot photofrin conjugates. So cells take these up, we think, by pinocytosis by looking at live cell trafficking in these. So it's no problem getting these into colonies of tumor cells, at least in vitro. And one can do the physics calculations to predict what dose of radiation would you need for a given energy transfer to get a given dose of singlet oxygen to be cytotoxic to the tumor. And if we do this calculation, we predict that at typical clinical doses between 2 and 5 gray fractions, that there would be a 30% increase in treatment effect relative to radiation alone. And the preliminary data in culture are showing this. So this is in a paper that's in review right now. If we look at either the enhancement ratio, which is a quantitation of the degree of tumor cell death relative to that achieved by radiation alone, we see an approximately 30% increase in enhancement of cell death after treatment in the presence of conjugates relative to radiation alone, and also notably relative to either quantum dots or photofrin alone. Second, we're doing clonogenic survival assays on tumor cell colonies, and we're finding that the survival fraction is significantly decreased when we do this in the presence of the conjugate. So the conjugate is acting as a radiosensitizer, and I should point out that both of these studies were done on A459 lung carcinoma cells, which are notably chemoresistant and radioresistant, and yet we're achieving some radiosensitization in these cells. So we think that we're on to an idea here that may or may not be clinically viable with respect to FDA approval of quantum dots for this particular conjugate, but an idea at least that allows us to achieve cytotoxic enhancement in the tumor cells simultaneously with radiotherapy instead of by a pre-sensitizing dose to the patient, in which we can use, especially if we put targeting molecules on here, the spatial cooperation with conformal radiation to achieve the geometric specificity to the tumor. And so, in general, these two are just 2 examples of work that we're doing to try and understand cell interactions with biomaterials from both a tissue engineering and a therapeutic point of view. And the idea is that, for example, in vivo, there are examples of microguidance systems in polymer scaffolds that the tissue engineers are trying to mimic in electrospun polymer scaffolds, for example. And so we'd like to be able to engineer both cell life and death with respect to tumor control or tissue formation by engineering these materials over the length scale that allows us to control cell-cell communication as tissues form. So there's a number of people that have contributed to this work, most notably collaborators in radiation oncology at UVA and nanotechnology, Robert Hull, who's now up at RPI in Albany, and Kurt Kolazinski, who fabricated the nanopillar substrates. And everybody has to put their own little nano profile of something they made, so there it is.